Start.
Yes, sir. A very good morning to all. Honorable Chancellor, Sir, Pro-Chancellor, Sir, Vice-Chancellor, Sir, Professor M.P. Pandey, Registrar, Professor Sanjeev Agarwal, Sir, Pro-Vice-Chancellor, Sir, Eminent Speaker of the Day, Dr. Sanjay Kumar Goswami, who is a Senior Scientist in Indian Institute of Sugarcane Research, Lucknow. Key officials of the University Administration, Director of SASE, Dr. Viren Singh, Sir, Additional Director, Mr. K.K. Bansal, Sir, directors of different school and participants of the webinar. Today, we all are here for a national webinar on agriculturally important microorganisms for sustainable agriculture. I, Ananya Singh, welcome you all to this national webinar. We all are very fortunate to have here with us today Dr. Goswami with us as a speaker. So, Dr. Goswami is a very renowned personality and in the field of plant pathology. And presently, he is working as a senior scientist in Indian Institute of Sugarcane Research, Lucknow. Dr. Goswami published more than 20 research papers in national and international journals and received PP Single Memorial Pesticide India Award in 2000 from Indian Society of Mycology and Plant Pathology. Dr. Goswami successfully handled 9 research projects sponsored by national and international agencies. We cannot summarize his journey and his work in words, yet he has achieved a lot in his academy. So, sir, I welcome you and thank you for being here with us today.
Thank you, Ananya. I am audible to all?
Yes, sir, you are audible. As we all know that microorganisms play a very significant role in our lives and in agriculture, to which we all are very closely associated. So basically, for an overview, the microorganisms in agriculture maintains fertility and improves the quality of the soil. It helps in composting process, which help in formation of manure. Microbes are present in soil to enrich the soil with nitrates and different nutrients. It also provides certain antibiotics to the soil and also some nutrient contents. There are few microorganisms which are used as biofertilizers as well. Rest will be told to you by Dr. Sanjay sir, but before that, I would like to invite Director of SASE Department, Dr. Virendra Singh sir, for his welcome note of the day. Sir, please.
Oh.
Yes, sir.
Good morning, everyone. Honorable Chancellor Sir Sri Rajiv Kothiwal ji, Pro Chancellor Mr. Abhinav Kothiwal ji, Vice Chancellor Professor Mahendra Prasad Pandey ji, Pro Vice Chancellor Professor Rahul Kumar Misra, Registrar Sir Professor Sanjeev Agarwal, Directors of different schools, Eminent Speaker Dr. Sanjay Kumar Goswami, dear participants. Myself, Dr. Viren Singh, extend a very warm welcome to Dr. Sanjay Kumar Goswami as a main speaker of the today's national webinar. Sir, really it is a matter of pride for all of us to have you today. Dr. Goswami is a very renowned personality in the field of plant pathology. Dear participants, today we are joined here in a national webinar, Important Microorganisms for Sustainable Agriculture. This webinar is in association with Indian Institute of Sugarcane Research, Lucknow. I hope that this webinar will be very fruitful for all of us. I am sure that this webinar will be beneficial for the students, faculty members, and all of. Dr. Goswami, you are requested to start the webinar. Thank you very much.
Thank you, sir, for the invitation. I extend my heartful greetings to the IFTMU University, Moradabad, and the School of Agriculture Sciences and Engineering. So, once again, good morning to all. And I thanks the Chairman Dr. Virendra Singh, Director SCSC, and Co-Chairman, Organizing Secretaries, and all the higher authorities of the university for inviting me in this webinar. So, dear students, I also welcome you to this webinar. I hope you will be benefited from this lecture. So, not taking much time, I will directly come to the my presentation. So, I am uploading my presentation. So, visible, my screen is visible now.
Yes, sir.
So, I think everything is okay. Screen is visible and I am audible.
Yes, sir.
Okay, thank you. So, let us start. In between, if you have any problem or interruption or any query or question, you can in between ask me, or you can also ask your queries or questions at the last of the presentation or in between also. You are most welcome. So once again, I welcome all and thanks for the invitation for this lecture. Thank you all. And as you know that our topic of presentation or discussion is that agriculturally important microorganisms for sustainable agriculture. You see that since the agriculture started from the time immemorial, we always depended on microorganisms for directly or indirectly. We have got many benefits from the microorganisms. Well, in the previous times, we were not aware of the importance what we were getting unknowingly from the microorganisms, but lot of benefits we have unknowingly got from the microorganisms. So, but now we every organisms, not all the organisms, because microorganisms they are in millions and billions species and genera they are available, and they all are not exploited and have not been characterized for their useful agricultural traits and their useful potentials and useful metabolites enzymes secreted by the microorganisms, which are directly or indirectly very helpful for the agriculture, and we can exploit and use those metabolites and enzymes for the agriculture production, increasing agriculture productions, plant disease management, nutrient availability, and other decomposition of the waste material. There are so many direct or indirect benefits we are availing from the microorganisms. These may be bacteria, these may be fungus, actinomycetes, endophytes. So many types of microorganisms are there on our earth, and hardly we have explored less than 1% microorganisms available on the earth. So, with the time, uh, we have seen that using so many chemicals and so many pesticides, we are finding that there are so many direct harmful or non-target effects of these chemicals on the earth and the microorganisms and on the humans as well. So, looking these things, now we have started more work, doing more work on microorganisms, and we are trying to use these microorganisms as a nutrients availability or plant disease management, and so many things. We will discuss one by one these things. First of all, I would like to brief what are actually the microorganisms. So, microorganisms, as I said earlier, or you know that they may be fungus, they may be bacteria, they may be other endophytes, actinomyces, cyanobacteria, and many other organisms, even they are protozoa, or so many microorganisms are available, which we have to characterize, and we have to find good traits from them for the use in agriculture. So, microbes are a common and an essential component of all the habitats on the earth. They are available everywhere. Metabolic versatility, as I discussed earlier, they are metabolites secreted by the microorganisms or enzymes, or different types of chemicals or compounds they secrete or they produce. Fungus also produce, bacteria they also produce, and those chemicals or compounds we can use for our benefits. We can use in agriculture, health sector, and food and other related industries. We have a negative impacts on the environment due to use of chemicals, as I said earlier. So you know that in the management of pests and diseases, we use a lot of chemicals. So we are finding the bad effects on the or non-target effects on the environment and human beings also. So now we are looking for the alternate measures or alternate any methods or measures for the management of our problems. So we are looking very much to the microorganisms, which we have found that they are very useful. So, by producing so much intensive or high producing by planting or sowing high producing varieties, the loss of nutrient is very high. That's why the soil fertility is continue declining, and productivity is a major global concern. So we have to fulfill the required production for the feeding of the increasing population. That's why we are using high yielding varieties, hybrid varieties, and we are using a high quantity of chemicals for the management of plant diseases and insect pests, and we are using chemical fertilizers as well. So all these things, yes, they are essential, and they are increasing the yield, and they are fulfilling our purpose of good yield, and that's why we have a food security because of these things. But because of this, now we are feeling some or finding some negative effects of these things. So here, microorganisms can play a very important role in improving soil texture, structure, and productivity. Uh, See, the soil is the very important living organism. You can say it's not a dead soil. Is not a dead. It has so many living organisms, bacteria, fungus, and many others. So, including plants. That's why a soil can give rise to a new young plants, then a big tree. So, soil we should never consider as like any dead structure or uh, anything which is non-living. It is a living thing, which we have to take care at utmost importance, because of soils we are getting each and everything. So a lot of microorganisms and variety and diversity of microorganisms it was used to find in the soils, but because of some change in the cropping pattern, cropping practices, the soil diversity is reduced in the previous many years, which we have to maintain it again by various ways. And microorganisms, they play a very important role in that mission, we would say. Uh, So microbes, why microbes? They are very important. Along with, I have mentioned here two books, very good books on the role of microbes in agriculture and microbes for sustainable agriculture. The students or any person, research scientist students, they can have a look for these very good books on the microbes and their use in agriculture. You will find it very useful. So Industrial agriculture requires increasing amounts of fertilizer and pesticides. I told earlier, this seems to be the result and of the poor cause of microbial diversity I mentioned before. Crop plants today lack many of their important symbiotic partners to produce or increase the contents of vitamins, minerals, antioxidants, and other metabolites that are beneficial for both plant and human health. So these are the setback, or these are the non-targeted effects of using these chemicals or pesticides, fungicides, insecticides. There are so many chemicals we are using in a huge quantity. Irrational use of the pesticides. All these things are responsible for this reduction in the microbial diversity of the soil or the plants and everywhere. Because these microorganisms, they are producing resistance also, and so because of these practices we are not able to sometimes we are not able to control these insect pests by the use of chemicals also. And at that place you can find the role of microbes where we cannot even control with the chemicals. Their microbes, they can be used as antifungal or antibacterial agents, biocontrol agents. They can be used for the management of those resistant microorganisms which they have produced resistance by the continuous selection process, uh, because we continue on using our chemicals, pesticides, these are things. So there is a selection process where some strong or variable strains come out in our environment, which we are not able to manage with our chemicals. That there comes the role of the microbes. So uh, uh, again, here you will see that soil is the ultimate source from which plants recruit beneficial microbes for the rhizosphere and phyllosphere. So maximum diversity and maximum microorganisms, they are found in the rhizosphere, and the variety and diversity, which is very important for any crop plant, we can say that is the diversity and different types of microorganisms, they are available, and different types of exudates from the roots and the metabolites or chemicals, they are released by the different microorganisms, and there is a network and strong interaction between all the microorganisms there in the soil. So there is a different ecosystem and different interactions. So many interactions are there in the soils or in the rhizosphere soils. So because of those interactions, they have make a balance. There is a natural balance of microorganisms in the soils. Because of that, the plants, they grow healthy, because the mycorrhiza and the other the mutual relationship between the microorganism and the plants. We know well known these things are. We know all these things. So plant rhizo, phyllo, endosphere microbes not only increase nutrient use efficiency and thereby crop yield also. Definitely they will make available those nutrients which are unavailable in the soil. These microorganisms, they dissolve or make the nutrients available for the soil. So then the plants, they cannot take every form of the nutrients of available in the soils. So the microorganisms, they convert the available nutrient into the in the form in which the plants, they can take it up. The nutrient may be available in the soil, but they are available in that form which the plants cannot take. So there the microorganisms, they come into play, and they make the nutrient into available forms, which will further be taken by the plants, and they can increase the yield, and those vitamins and whatsoever we are getting from the crop products, that is useful further for us, and that will be free from the high pesticides and other residues which we don't want to see. Now, because we also export our materials, our food grade productions, either fruits, vegetables, and so many things. They are all agricultural produces. We send to the different international markets. There is also we have a certain residue limit. We can't cross that limit, so we have to keep the residue at a minimum level to have that to enter into that international market. So that's uh, that's where the microbes we start using to produce the crops organically producing the crops. So there is also. So we will discuss one by one many things here in organic farming, because now there is a very much there is in every country they are demanding or producing the crops without the use of chemicals, whether chemical fertilizers. We are trying to reduce chemical pesticides, herbicides, all those things we are trying to reduce, so that we can have a minimum, or we should not cross the residue threshold residue limit, so that it should not be harmful for the humans, because directly or indirectly we are taking some amount of minimum amount, or we can say that sometime increased amount of pesticide slowly and slowly, which with the time in 1 year or 2 year 3 years, which continuously accumulates in our system, and which will be further harmful for us. So those are the things we have to avoid. So pest management in organic farming, which you know that in organic farming we mainly use the some organic produce, and we avoid the chemical fertilizer pesticides. So organic farming is gaining wide attention among farmers as well, entrepreneurs, policymakers, and agriculture scientists. Disease management in organic farming begins by taking the right decision at right time. That organic farming is all about changing all the agronomical practices, like sowing time, selection of seed. We have to select a healthy certified seed, and then the sowing time matters a lot. By changing our sowing time, we can escape the disease, we can escape the insect attack. So, the place of the sowing area, agricultural area, we can shift crop rotation is there, and the altitude of the our agricultural land, and then there are many other practices, economic practices are there. We can use resistant varieties. There we have to use very less amount of chemicals. So there are so many things in the organic farming which we have to take care, and one of them is microorganisms. At least you see the application of biocontrol agents can help to reduce diseases caused by pathogens like armillaria, Fusarium, Sclerotium, Rhizoctonia, Verticillium. You see the Fusarium, you will find Fusarium and Sclerotium in almost all the plants. So you can say approximately all the plants. Rhizoctonia also in almost all the plants. These pathogens simply destroy the root system of the plant, and further you will not get anything. It may be pre-emergence or post-emergence. These pathogens, they're very harmful, but these pathogens, they can be used, they can be controlled by the use of very good control agents like trichoderma is there, and some non-pathogenic Fusarium species itself, they control the Fusarium, and there are pseudomonas is there, and in the bacteria, if we go to the bacteria, there are bacillus, and there are so many strains now. They are available with the institutes, or not only with the institutes, or with the in the markets. In the markets, we can have very good biocontrol agents. So slowly and slowly, we are moving towards the organic farming, because it is not like that we can shift to the organic farming or chemical free farming within 1 year or 2 years. Slowly and slowly, it will take 4 or 5 years. Because if you have to go for organic farming, you have to wait for 3 years at least. You have to keep your land free from chemicals, any types of chemicals for 3 years. Only after that you can go for organic farming, because there are residue. If you have earlier used chemicals, then there will be a residue in the your field. So we have to go for or wait for 3 years to make it free from the residue. So uh, there are so many fungus or fungi and bacteria they are available to manage uh, each type of pathogens. Here I am not mentioning the detailed list of those microorganisms, but in between all the slides there will be few examples where you will find that these organisms can be managed by some particular type of useful our microorganisms. Pathogens, there are so many pathogens, and there are so many pathogen microorganisms are there which are useful for the decomposition, and decomposition will further convert the residue, crop residue, into some important uh, our compost or other type of manures which we can use for our field, and we can avoid the use of chemical fertilizers. So nowadays, many entrepreneurs and policymakers they started and they are putting a lot of funds into the organic farmings, and even the government of India, they have a lot of funds for the organic farming, and they are encouraging and giving funds to the startups, and many institute and universities, they are being encouraged for the organic farming, because it is a slow process, and it is not a quick and shortcut methods there for organic farming. We are listening to organic farming since many years, but still we have not at that stage where we can produce a sufficient amount of food grains or eat our vegetables and fruits that can meet the demand of the our acres in increasing population. So still we are dependent on the chemicals, but slowly and slowly we will reduce the use of chemicals, and will move on to the organic farming without using the chemicals. So disease management, I have told that microorganisms, they are maximum being used in the disease management and in insect management also, like bacteria and some many other ready-made formulations which are used for the management of insect pests on the sites. We are listing some formulations which are available for ready-made available with the institutes, and in maybe the same formulation may be available with the markets also. So eco-pesticide is one of the very good formulations having pseudomonas fluorescens, and eco-green fungicides and other formulations that contains trichoderma viridie. Green fungicides, trichoderma hertzianum, it has the active ingredient, are effective against a number of soil and seed-borne pathogens. Like you see, these two or three formulations, they are effective against the pathogens like Rhizoctonia, Sclerotium, Sclerotinia, Fusarium, Pythium, Ralstonia, Macrophauna, Bipolaris floma are the major genera of bacteria and fungus which causes major diseases. And the only three formulations, they have covered so many pathogens which we can, and they are highly effective against these pathogens, these formulations. Biopulse is another formulations, but which contain trichoderma hertzianum and bacillus amyloid coefficient is effective against Physidium wilt in chickpea, and not only in Physidium wilt, and in other wilt diseases, it is very useful. So you can have these formulations from the National Bureau of Agriculturally Important Microorganisms. They have these formulations, and you can conduct some experiments, and you can have you can design your experiments like that to see the effect of these biocontrol agents or these formulations in your field trials, also in your pot or polyhouse conditions. Starting from the lab, you can first test it in lab, and then you can conduct or you can design your research for looking at the efficacy of these biocontrol agents, which will like that it will spread or spread the information, and everyone will be able to see the result which we will find out of it, and slowly we will be able to extend the information, the research products to the different universities, different farmers where it will be tested. And the purpose of research, ultimate purpose of research is to take the products from the laboratories to the land, the farmers' lands. That's why I am mentioning these products and mentioning the source as well. That there are some others formulations are also there. Pseudomonas fluorescens based biopesticides formulations, and they are effective against the abiotic stress management. Also, there are many abiotic stress problems like soil salinity, temperature issues are there, and some flood conditions, some drought conditions there are there. So in those conditions, for those problems, many institutes they have developed the formulations to manage those issues. Like we can produce a good crop in the drought soil by using those microorganisms, because those microorganisms they have a mechanism or mode of action which make the less available water to the plants. Actually, in the drought soils or the where their irrigation problems are there, the moisture level is also there, but that much moisture level is not in the available from the plants. So these microorganisms they have such mechanism or such type of effect that also make the less available water to the plants, and they survive in the drought conditions also. Pseudomonas aeruginosa and this geniculate MF84, they have also antagonistic activity in Rhizoctonia solanae. You can use all the students, they can design their experiments, design their research on these formulations also. It will be good if they will test it and they will find some good results from it. Actinomycetes, Streptomyces sarbiensis, and Mycolibacterium pulver is found effective against false stomatophorus, which is not main, also controlled by the uh, canicles. Also, even canicles are very less effective against this false stomatophorus. They are also we can use these microorganisms, useful microorganisms we can use. So there are so many other organic options. Uh, there are species like Trichoderma, Glycoplatum. These are the main genera or species which are being used in today's days. Pseudomonas bacillus is a single genera which have so many formulations available. It's a bacteria which is being used for so many places. For the insects also, like the cotton borer, for the cotton borer it had been used. You all know that it turns uh, the gain towards the human agriculture by producing a lot of cotton production. Because of this Bacillus thuringiensis we have used in cotton and genetic crop. common scab of potato, it's not does not cause huge loss, but it has some aesthetic value in the market. So it's a bacterial disease which is also we can manage by using some saprophytic species of streptomyces. So that will take care of this disease, common scab of potato. So common saprophytes fungus Trichoderma viride is able to parasite the mycelia of the fungus. It is we all have this information. There are other formulations on castor. It is a resulted in 98 to 99% decrease in population of this Cania geneta and Spodoptera luteura. These are the Spodoptera luteura. These are the insects which are found in the castors, which are managed by these formulations. I mentioned BOR BT127. Sunflower, again, it is very useful against many pathogens, and in combination with this, the Beauveria bassiana is also very effective. And Pneumonia relay and Beauveria bassiana, I already mentioned, these are very good and very effective against the insects. Liquid BT formulations, we have discussed it, it is very good against the borers. Uh, like uh, we have mentioned, the Bacillus thuringiensis is effective against many types of weather, whether it is cotton or chickpea or any other crop. So this you can see from the slide, and this Bavaria bassiana and Metarhizium is another very important genre, and this Bacillomyces, they are very effective against the sucking pest in vegetable crops. Any student, those who are working on the vegetables or the fruits, they can have these formulations, or if they have not available these formulations, they can simply uh, isolate metallizium, or they can have it from some culture collection center. From there, they can have it, Bavaria bassiana or metalliziums. They can check it, or they can use it for the management of these sucking pests in their trials. Also, they can use it. Entopathogenic fungi are also there. They have broad spectrum promising strains. There are so many which are effective against different diseases of the many different crops, and they are able to increase the yield by by manifold. Which, if they will not use, there will be a huge loss, and these biocontrol agents, or these good fungi, you can say that, will be very useful, and we will not be having you to use the chemical against them. We can prevent these diseases and insect by using these useful fungus bacteria, or you can say that antifungal or antibacterial bioagents, which now we have available. Uh, and they are available with different institutions. You can have it from the institutes, different culture collection units, microbial formulations for nutrient management. They are also available, and here you can see that Bio NPK, Bio Grow. I have given the photos also, and the samples photo of the. These are the liquid formulations, and the all the method how to use it is itself available with the label of the product. You can use these formulations, or farmers can also, students or scientists, they can conduct a trial by these formulations in the farmers field, or there they can see the results, and how these formulations, they can decrease the use of chemical fertilizers, and we can replace the huge quantity which we used uh, for fertilizers. We can replace chemical fertilizer with these microbial formulations, which will make available nitrogen, phosphorus, and potassium. the Azotobacter crucocum and Tannibacillus stylophyllae and Bacillus decoloration tennis. And these are the microorganisms which are useful, and they are very good, important active ingredient of the bio-NPK and bio-grow, as well a consortium of some solubilizing bacteria, phosphorus solubilizing bacteria, as hydrophore and IA producing bacteria, indole acid, which is helpful for the growth and some healthy growth of the plants, because these some of the microorganisms, they does not prevent the disease or any other pest, but they have healthy growth of the plant. So that healthy growth itself avoid the pathogen infection or pest infection, because if the plant will be healthy, there will be less infection of the or less attack of the pathogen or pest, you can say that. So in one trial, 25 to 30% increase in yield was recorded in a treatment inoculated with BioGrow in tomato crop. So you can see, without using any chemical, we reduce the 25 to 30% yield loss, and that is enough. That is a very good result. That if you can increase the yield 25 to 30% as compared to control. In control, if as compared to control, if you can increase the yield 25 to 30%, that is a huge benefit, and there is a huge, you can say, huge impact on the crop. So slowly and slowly, these types of formulations will be available for us, and we will be able to use these, and continuously, slowly and slowly, we will reduce the use of many chemicals. So likely Biofos and Biofos Plus are liquid formulations, and phosphorus solubilizing bacteria, they are there to make the phosphorus available. So because the Indian soils, they are deficient in phosphorus soils. So by using they are actually not deficient in phosphorus, but the available phosphorus is less. So by using these formulations, I have given a picture along with my presentations, you can see. So we can use these formulations. So this is a lot of information, lot of formulations now we have available for each and everything, like either it is a biotic stress or abiotic stress. For each type of problems, we have this microorganisms formulations, they are available. So biopriming, it is a very important another aspects. We can uh, with the seed coating, like seed coating, the microbial, we have a microbial consortia which we use like a seed coat. We make a paste of those microbial formulations, and we mix the seed of the particular crop, and there is a seed coat of microbial layer, microbe layer. That seed coat, when we put that seed into the soil, then there is a colony of that useful microorganisms which take care of the seed starting from the germination till the harvesting stage. So, and those useful microorganisms, they increase their populations in the soil as well, sir, and many types, different types of benefits, starting from the nutrient availability, managing diseases, pre-emergence diseases, which are caused by the Tritium, Phytophthora, Rhizotonia, and then post-emergence diseases. There are so many diseases which come after post-emergence, according to the climate and according to the crop host. There are different diseases which are managed by the biopriming and seed. If we will be able to make the seed or having a coating of a such type of microbes which can keep away the pathogens. So at that stage, we will be successful, and it is a very good beginning. That will be very good beginning if we will be having it. Starting, we will start from the disease-free environment, then our further plants, our further crops will be very good. So seed treatment, it is very easy, and suppose we have 8 to 10 kg of paddy we have to sow. So 8 to 10 kg paddy we can treat with some cyanobacteria, which will make available different types of nutrients, and other formulations are also there which make nitrogen, phosphorus, all other nutrients available. We will prime the seed of 8 to 10 kg. That is easy. Uh, to give the treatment to the seed, 8 to 10 kg seed, instead of giving treatments to the field of large area. So all the formulations, they are good treatment at the seed level instead of going to the field. But we have a formulations and treatments, they are available for at the field level also. But those treatment of formulations, they are not so practical, and it will be difficult to follow those practices in the field where there is an area of 2 3 hectare or 4 hectare. So at that stage it will be difficult to manage. So it is better to go for biopriming of different seeds, different crop seeds with different types of formulations for different management practices, whether it is nutrient management, pest management, whatsoever is the problem. For every problem there is biopriming available for different seed crops. So this is biopriming is very important in organic farming or chemical-free farming, because if we are going to see the seed of say mustard, we will need a very small amount of seed for 1 acre to treat 3 kg, 4 kg seed maximum we will need. So it is easy to treat the seed at that stage instead of giving the treatment in the field. So this is the best stage, and this is the best to treat the seed itself rather than going for the crop treatment. See, the zinc, bio-zinc is also available in rice. There is a Khera disease, which is because of unavailability of the zinc. Some places it causes huge loss to the rice disease also. Similarly, bio-potash, genocell, there are so many formulations I am mentioning here. So the details you can, though I have presented my details here also, you can have more information later on from me as well, or as well from the concerned institute, that is the National Bureau of Agricultural Institute from Mauna Kanchan. That is the place name, and you can go to the website, and there are some other cultural collections. There are many cultural collections in different places, like Indian type cultural collections in Delhi, and microbial type cultural collection in Chandigarh, and Agarkar Institute, they have a cultural collection in Pune. So there are so many cultural collections uh, which provide these microorganisms, which we can you also can develop your own formulations by using combining 2 or 3 useful microorganisms. First, you have to go for the traits. You have to find out the important traits, like what are the important traits, whether they are the phosphorus solubilizers, or they can manage some disease, or they are nutrient availability they form, or they are go for the composting. So, by taking these microorganisms, useful microorganisms, you can make your own consortia, or you can make your own formulations, and you can go for patent, go for copyright, and that will be product will could be a product of your institute, and you can use these products, or you can go to the some company, they can use it. That will be of their benefit, and definitely it will be a good for the farmers as well. So these things, these are the well trust areas, or some upcoming areas. Every institute or every research students should go for it. And there are some more examples, like cyanobacterial consortia, and Bradyplasm japonicum is also there. They are useful or they are very good in increasing of different crops. Uh, these are the findings of the long-term research and long-term trials, repeated trials. These are the findings from that places. So you can see the potential of agriculture important microorganisms. They have not microorganisms. They have not left any field unaffected. In every field, wherever you go, they have their roles. They have their very good and very revolutionary rules they have, but we have to identify and characterize. Uh, we have to exploit those microorganisms, those useful microorganisms which are highly useful for the agriculture of India. So that is that type of research we have to do more and more. And uh, see that the microorganism availability and diversity of microorganisms is such a large diversity and such a large number of microbes they are available because we will not be able to uh, complete 2-3% of the diversity. So more and more you will explore it, more and more you will explore all the plants, all the soils. So you will keep on finding novel and novel um, useful microorganisms, and that will be further you can incorporate into our agriculture system, which will give further a very good yield. And microbial consortium for composting, you know that we need some cellulose breaking organisms. Here, biocompost consortia or formation is available. For their lignocellulosic fungi are there, Trichoderma viridis, there Aspergillus niger is there, Pleurotus florida, and Phanerothene Chrysostorium is there. These are being used for the composting of lignocellulosic waste. So it is very important. We cannot imagine life without fungus or bacteria. Suppose if there is nothing to decompose the waste material of the earth, then the whole earth will be turned into a garbage. So these microorganisms they play a very important role in converting the waste material or all the garbage into a good uh, manure which we can use into the field. If you visit any field, you will see there automatical conversion of the waste material, the crop waste material, or there is a animal waste material and all other waste material. They collectively they turn all the things into the important this compost or some good manures. So we can enhance or make this process very fast by using or by applying the particular microbial consortiums which are available with us. In the nature, there is also a composting process, but if we want to enhance this process, we can use the available consortium. With in the literature, there is so much information is available with the institutes, and there are many private public sources are there from where we can have information on these, and you can also explore many micrometers for different useful activities. So that could be a very good part of your research, and we can enhance the pace of our upcoming mission where we want to turn into organic farming. So PGPR, it is well known that plant growth promoting rhizobacteria. It is growth of any if there will be a good growth of any plant, so there is always a less chance of any disease or insect or pest attack. Even if there will be attack, then the these plant growth promoter they compensate the loss which is caused by the insect pests. So their plant growth promoters they play their roles, and it is also very explored areas and a very researchable area, and a lot of research is being continued or being done on the plant growth promoting bacteria. Biocapsule is the one formulations, dark based formulations are also available which are managing. Um, many problems of our agriculture fields. So Bacillus megaterium based formulation is also available, and at which we use at different places and different against different problems. So this there is unlimited information available. I cannot put all the things into one presentations. And but as I mentioned, the two books earlier, and there are more available also. And you have to contact some institutes, or you have to isolate your own good, useful microorganisms, and you can test those microorganisms for different useful activities in the agriculture. So these are the things I am presenting before you, just to ignite or just having giving you a spark that how microorganisms they can turn the things around for the human beings. Means any impossible work which is not possible by the humans, which is not in the hand of humans, the microorganisms is tiny little not visible organisms they can
बस 5 मिनट के लिए रुक जाओ बोलना है ना मुझे 5
So, in the abiotic stress, we have, I have mentioned earlier that they play a role in the abiotic stress in the soil salinity. There are so many examples I have mentioned here which play a role in the soil salinity. You can see that the catalase or superoxide dismutase and peroxidase. These are the some reactive oxygen species and which act as a defense, and pseudomonas ACC deaminase producing organisms, then trichoderma longibacterium, the IAA ACC deaminase producing. So there are so many organisms which produce catalase, superoxide dismutase and peroxidases, which further uh, uh, reduce the effect of the salinity. So abiotic stress management here, there is one picture is there. Just a moment, one call is here. Hello. हाँ जी. हाँ हाँ बताइए. हाँ ठीक है. क्या चीज? अच्छा अच्छा मैं करवा दूँगा. Here you can see that there are abiotic stress management. I am trying to make you clear by one diagram. You can see there there are root exudates are there, and then the factors shaping the plant microbial community. And endosphere microbiota is there, phyllosphere microbiota is there, root exudates, root rhizosphere is there. So there are so many. You can see the small blue, green, or red colors. Some figures are there, which has been used as an bacteria or many other things. So it can be used at the places of like deserts where nothing is being grown because of the lack of water or some other things. So like there is a low precipitation is there, temperature is very high or fluctuating temperature is there, high solar radiations are there. So at these places uh, we can use these microorganisms in a better way, and there are some microorganisms they can tolerate the temperature up to 50 degrees Celsius also, up to 60 degrees Celsius also, or even more than. So we have to isolate those important microorganisms from the hot springs also, from the places where there is a high temperature. So So these are the things how we can change the desert into some green area, because we have available so many things, so many microorganisms who can turn the barren land into a good agricultural land. We have to start from a small piece of area, not from like a very big area, but from a small area. So that's how we can do it. And here another picture is there. You can see that there are rhizospheric microorganisms are there. There may be fungi, bacteria, and many endophytes, and herbus micro rhiza is there, and cyanobacteria is there, rhizospheric soil is there, plant roots are there, salt will be available there, and so all these abiotic stresses, if there is a salt is there, and then there are so many bacteria and there are some useful microorganisms who by different mechanism, I have listed the mechanisms above, like production of IIA, CAT, COD, POX, ACC, deaminase activity, like these, so many activities are there, which will reduce the effect of salt stress, because there are some bacteria who can tolerate the salt salinity up to the highest level, and uh, so they have that mechanism. Uh, they have some chemical or some uh, metabolites which secretes and which act as a shock absorber, high salinity, the shock absorber, which will prevent the salt stress management. So I'll not discuss much in detail. You can maybe having these slides. Maybe later on I will share it with you, so you can uh, take it with you. So these are some other informations on the salinity stress management, drought management. So examples are given there. You can have these things. So the future outlook is that if organic farming systems are to deliver substantially amounts of food to future food systems, and at the same time deliver multiple other benefits, as is the ambition according to the organic principles and as it is expected by consumers, the organic sector and its producers, breeding companies, advisory services, farmer associations, and the public policy all need to focus on a broad set of goals. Means it is not a single person's work; it is a team work. So many organizations, so many entrepreneurs, so farmers, breeders, and many more. They have to come together, and they should have some proper policy, some proper committees, some proper societies to take care of all these things. That how we have to proceed in the organic farming, the farming without chemicals. So we have to have a proper plan and strategies to proceed further. So there are some take-home messages that we can use. We know that there are many non-target effects of chemicals, so be aware of these things. Organic farming is the need of the time that we know, and microbes will make it possible for humans to practice chemical-free agriculture. We can explore and utilize the potential of microbes. So there are unlimited, uncountable numbers of useful microbes available in our environment, in our diversity. We have to explore and find it out, and we have to use for the benefit of the agriculture. Achieving food security for the growing world population requires scientific breakthroughs and technology developments at many fronts. Enhancing soil productivity through better resource availability and use efficiency in arable soils is one of those key challenges where microbes can play their important roles. So this is how microbes are important. Success in microbe-based agriculture will not only improve farm productivity and profitability, but improve soil, air, and water quality, which are very important for the human beings. We cannot think, we cannot imagine a life without good quality soils, good quality air, fresh air, and good quality uh, and pure water. We always need all these things. So microbes, they play their role in these things. So we have to have move a step ahead. Together, we can be able to achieve our goal. And this is a brief presentation from my side. A lot of information is available, but a little spark or just a sensitization type of presentation I have given so that we can have some information that what is available and what more we need to have. So with this, I thank all for listening the presentation patiently. Thank you all for inviting me. Thank you very much.
Hello,
Thank you
Ananya
so
ma'am.
much, sir. Yes, sir. Am I audible?
Yes, ma'am.
Okay. Thank you, sir. Thank you so much, Dr. Goswami. On the behalf of all the participants and IFTM University, sir, your lecture was very informational to all of us, and I'm sure we all must have got benefited from it, or we all must have gained some of the other knowledge from it. So thank you for your speech today. It was very informational.
Thank you very much. Thank you. Thank you.
Now I would like to call Dr. K.P. Chauhan, sir, HOD of Agricultural Sciences, School of Agricultural Sciences and Engineering, for vote of thanks today. Sir, please join us and deliver the vote of thanks.
Namaskar, sir. Honorable Chancellor, sir, Pro-Chancellor, sir, Vice Chancellor, Professor MP Pandey, sir, Registrar of IFTMU University, Professor Sanjay Agrawal, sir, Eminent Speaker, Dr. Sanjay Kumar Goswami, Senior Scientist, Indian Institute of Sugarcane Research, Lucknow, key officials of the University Administration, Directors of School of Agriculture Sciences and Engineering, Dr. Virender, sir, Additional Director, Sri K.K. Bansal, sir, Directors of the different schools and participants of this national webinar. Remembering the Almighty of God, I present my sincere thanks to our university administration for organization of one-day national webinar on agriculturally important microorganisms for sustainable agriculture. I am also thankful to our eminent speaker, Dr. Sanjay Kumar Goswami, Senior Scientist, Indian Institute of Sugarcane Research, Lucknow, who accept our invitation in a short notice and proceed the consent in his very tight schedule. In present scenario, our agriculture faces several challenges. Likewise, degradation in the soil health, untimely and uncertainty of climatic conditions, costly inputs, and post-harvest losses, etc. To overcome and neutralize the negative effect of these conditions, microorganisms play important role. I think the topic of this national webinar is very fruitful to our participants. I am also thankful to organizing secretary, convener, and all other persons who are directly and indirectly involved in this. I wish you all good luck. Thank you. Thank you, sir.
Thank you, sir. Thank you to all the participants whoever joined here and whoever attended the session today. Thank you to all.
Now you may leave, sir.
Okay. Thank you to all.
Okay.
Thank you.
Sir, namaskar.
No, thank you.